I'm speaking with Helmy Eltoukhy. He's the co-founder and CEO of Guardant Health. Oh, Helmy, thank you for joining us.
Yeah, thank you, Bambi, for having me.
I want to start with a 50,000-foot view question. What have been some of the technological breakthroughs in cancer treatment over the last 5 years?
Yeah, that's a great question. Cancer is an area that, there hasn't been much progress since, recorded history for thousands of years. You even look at cancer death rates for the last, 30 to 40 years. They haven't improved very much, but the last 5 to 10 years have been very promising in terms of the treatments that have come out. So the one area that has been a game changer are targeted therapies. So these are drugs that target specific defects in the genome that cause cancer to be cancer in patients. So you'll have certain genes that are just defective that cause those cancer cells to grow and multiply much more rapidly. And so we've come up with, the pharmaceutical industry has come up with these targeted therapies that can actually specifically, surgically strike those defects in those cells and kill the cancer cells much more specifically than old standard cytotoxic chemotherapy. So we used to just bombard patients with mustard gas and stuff like that would just hopefully kill the cancer cells faster than it kills our normal cells. And that was the first class of chemotherapy we had. The second class targeted therapy is much more like a smart missile, a smart bomb that can go in, just target those cells. The side effect profiles are much, much better, just typically rashes or very slight side effects rather than these horrific, adverse effects that cancer patients would have to normally go through. And then now, over the last 2 years, there's been a new class of therapies called immunotherapies, where they essentially are able to employ and exploit our immune system to go and help fight against the cancer. So that is the latest and greatest and most exciting development right now. So on the diagnostic side, one of the challenges we've had is, we have these targeted therapies that are out there that are genomically profiled, but how do you know, which genomic defects there are in cancer patients? Right now, or before Guardant, the best way to do that was to cut a piece of tissue out of the cancer patient through a biopsy, and then sequence that on a DNA sequencer to find out, what was unique from a genomic level in terms of those cancer cells. Unfortunately, though, there's a big inertial barrier with targeting, with actually performing these biopsies. The average lung cancer biopsy costs $14,000 and has a 19% complication rate. And so these cancer patients, are sometimes, hospitalized or dying not because of the cancer, but because of these biopsies and these surgical procedures, just to try to access the genomic information about the cancer or other information about the cancer. So, what we're really excited about is what we've launched about a year and a half ago in June of 2014, which is a liquid biopsy. It's essentially a way of targeting and finding out what those genomic alterations are through 2 teaspoons of blood. So much, much cheaper, no complications. And so we're essentially, it's almost like wireless technology versus, corded, phone technology in terms of the implications of what we can find, how we can follow, how we can monitor these patients in a much more humane and much more, kind of routine manner than we used to.
Give the analogy about the camera and the pixels in terms of, you and I spoke before, and you brought up that liquid biopsies, or at least looking at these CTC cells that have been around since the late 1800s.
Yeah, we're really standing on the shoulders of giants, is the way I put it. The first circulating tumor cells. So there's multiple ways you can access the genomic information in tumors without doing a surgery. One is there are whole cells called circulating tumor cells that kind of slough off these tumors and circulate around the blood. The first CTC was discovered in 1869, believe it or not. So it's been almost 150 years that people have imagined we could do something like this. What we look at are CTCs. We look at basically shed DNA that's this detritus DNA, almost waste DNA, that's being shed from these tumors as they turn over. That's called cell-free DNA. And that cell-free DNA was first discovered in 1948. So also been almost 67 years since We've known about these biomarkers. The nice thing about cell-free DNA, there's about 100x more tumor-derived material in a tube of blood than CTCs. So it allows us to get a very rich and complex profile of the cancer, not just at a single location like you do with a biopsy, but essentially the entirety of the disease within an individual. Many of these late-stage patients have multiple metastases, multiple tumor sites. How do you know which one is actually going to kill the patient? And each one of these may have a different genomic profile or a slightly different genomic profile that may respond differently to different targeted therapies. So when you make a choice to do a biopsy, you're making a choice to only look at one small part of a patient's cancer, versus what we see is essentially a superposition or a kind of a sum total of all the genomic alterations there are in that individual.
When you do a biopsy, a tissue, you take it from a certain part of the body, but when you take a blood sample, how do you know where that's coming from, or how do you identify where that tumor is in the body?
So we don't know where it's coming from, but what we see is basically a sum of all the genomic alterations that are being shed by different parts of the tumor. So what that allows us to do, it's very quantitative. So we can say that we may not know exactly that this tumor is has this gene mutation, this one has this one, but we can say that this patient is 90% this genomic material, the genomic mutation. They're 70% this one, 15% this new one that's coming up, and so we should be watching out for that. And so that's really what we're bringing to the picture with liquid biopsy, is this concept of time. Right now, cancer patients are being treated on the basis of old information from a tissue biopsy they may have received or obtained at diagnosis 3 or 4 years ago. Nowhere else in medicine are we treating patients based on old data points from a few years ago.
Well, you mentioned that that liquid biopsies and looking at cell-free DNA that sort of was discovered in circa 1950.
Yeah.
But talk about what's happened in the last 5 years that's enabled you to actually advance.
That's a great question. So I've been working in genomics since 2001, since the first genome was sequenced by NIH and others, and that genome cost anywhere from 100 million to a couple of billion, depending on how you do that counting. And when I left Illumina just about 3 years ago, the genome cost about $1,000. So that was really the first ingredient, the enormous decrease in cost that happened over the last decade in terms of sequencing a human genome. We just couldn't generate that information fast enough, cheaply enough for this to be feasible. So that was the first breakthrough that happened, basically as a result of many, many different players in the field working to make that happen. The second, though, was even these off-the-shelf technologies that exist today. You buy a sequencer from, some of the big names. The error rates are too high to be able to see these tiny concentrations of fragments in a tube of blood. And so we had to improve these error rates. We had to improve the accuracy by 1,000-fold for us to be able to see with very, very high quality these minute, concentrations of DNA. It really is like going from, a grainy black and white television to your 4K TV today, or kind of digital cameras from 10 years ago to what we have now. We had to essentially build that kind of improvement in the technology for this to be possible. And that's what's proprietary about Guardant's technology, is essentially our ability to both improve the sensitivity by over order of magnitude, but more importantly, improve the specificity or the accuracy by almost 1,000 to 10,000-fold. And so that really is what has enabled us to employ this technology on a...
So you stand by that your testing will be able to uncover and see more granularly than other tests?
Yeah, it's like almost like, if you need glasses and your vision's 20/200 and you can't see the letters, that's kind of what using kind of current sequencing technologies on blood samples gives you, and we essentially get you 20/20 vision on that.
And just to confirm, this does not detect if you have cancer. This is just to talk. This is if you have cancer already. This is to show you the progress.
Yeah, this is for this product, Guardant 360, which we launched about 18 months ago. is really for late-stage patients. So about 1 million patients in the U.S. that currently have cancer. Their treatments may stop working. The physician is wondering what's going on here. Instead of going back and doing another biopsy in these patients, we can just take a simple blood draw and stay ahead of the disease. Mm-hmm. The nice thing about the technology and why it's so profound, at least we believe, is that as we improve the sensitivity and sequencing cost continues to come down, mm-hmm. we can then target the 15 million cancer survivors out there. They're obviously, right now there's no quantitative detection for these patients for recurrence. Right. Like I'll give you an example, a breast cancer patient who's had a double mastectomy has an 8 to 9% chance of recurrence in their lifetime. And right now, the standard of care is symptomatic detection. So they ask them how they feel every year. Do they have back pain? Are they coughing up blood? And unfortunately, that's how we detect recurrence in those patients, which is really not where we should be in the 21st century. And then as we continue to make the technology cheaper and more sensitive, we can then think about why we started the company, which is essentially that test at an annual physical that every one of us can take and hopefully detect cancer stage 1, stage 2, rather than stage 3 to 4, like many patients are blindsided by today.
And just be part of the annual checkup cost.
Exactly.
How much does it cost today?
Right now, the list price is around $5,000. You compare that to doing a biopsy, which is $14,000. Getting the biopsy is just getting the tissue. You still have to do genomic profiling on top of that, which can cost another $5,000. So it's basically $5,000 versus somewhere between $15,000 to $20,000. So it's obviously much better side effects profile. A simple blood draw, Band-Aid is the worst complication, and much cheaper for the system. So it's a nice value proposition.
How do you measure the success of the efficacy or the efficacy of that test? For instance, you have a late-stage cancer patient who decides to take the blood draw versus a tissue or surgery for a biopsy. Yeah. How would you know unless they did both?
Yeah, so we've done large clinical studies, concordance studies or clinical validity studies where we've taken now hundreds of cancer patients. We've gotten tissue biopsy from them, sequenced
hundreds.
it.
So hundreds.
Yeah.
Tens of hundreds or?
No, I mean it's probably approaching 1,000 right now in total. With the first of those studies was a 165 patient study that we published in PLOS ONE a couple of months ago, and so that's all publicly available. People can take a look at it. And then we've had other external validation studies by other cancer centers where they've given us blinded samples. They've sequenced the tissue and they're testing us. And what we found in these studies is neither tissue biopsy nor blood gets 100% of all the mutations. Everyone misses a little bit. So we get 85% of what you see in a tissue biopsy. Tissue biopsy only gets about 80% of what we see. So we actually counterintuitively see a little bit more of the patient's cancer than a tissue biopsy. But in terms of where we match, we're 99.3% concordant. So our diagnostic accuracy is 99.3%. So, if we see it, it's really there. So
and it's cheaper.
And it's cheaper and it's less invasive. So that's the reason why this test, since we launched in June of 2014, to our knowledge, has been one of the fastest growing tests in, this segment of the market. We're running more than 20,000 samples a year right now, which, is something that is, not been seen in this field.
What does that mean, running 20,000 samples?
So 20,000 patients are, taking, a year taking Guardant, 360 tests. And, I think to put that in perspective...
Oh, I thought you said there was only 1,000 patients. I'm sorry.
No, that's part of the study.
Oh, okay, for the study.
For the study. So that's under a rigorous kind of study where we're getting tissue biopsies from these patients and looking at the data. In between the two. But in terms of actually running the test commercially, clinically, and we're in 20 out of 24 NCCN centers. So most of the top tier centers are using Guardant 360. We sequence more patients than either genomically than either Memorial Sloan Kettering or MD Anderson. And I believe we sequence more than both of them combined which some people find surprising. So
and for one specific patient who would typically do one tissue biopsy a year, or even every couple of years.
Well, sometimes in their lifetime. Most of them are just once in their lifetime.
Your typical patient now, are they doing a couple of liquid biopsies a year?
Yeah, it's just starting to come up. It's a new paradigm of being able to watch these patients, over time. So when we started, very few were getting repeat Guardant 360 tests. Now we're approaching about 15% of our volume is serial. Are doing it serially. So it's been really exciting to see, because these drugs out there are still limited, right? There aren't very many choices for these patients. So the drug may work for a while, but eventually it stops working. And whenever the physician has to ask that why question, why is this drug not working, and what better drug can I put this patient on, that's when they really need to do a biopsy to see why the cancer has changed at a genomic level and why it's not responding anymore. Right. And so that's where another Guardant 360 test is appropriate. Got it.
But you still have to rely on the judgment of the oncologist.
Yeah, it's
the availability of the
drugs. It's a learning, process, right? It takes 7 years for 50% of physicians to change their behavior on average. That's the half-life that's there. And so the fact that we're at this point where so many patients are having their first Guardant 360 test is something we actually is beyond our expectations right now.
And so these are late-stage cancer patients, right?
That's these are all late-stage.
And there's about 1 million of them. Is there a certain type of cancer that that works better?
Yeah, so a large percentage of our volume is lung cancer, breast, and colorectal, so the 3 main ones that you'd expect. We have good detection rates there, and there's more you can do from a genomic level in terms of targeted therapy. So lung cancer, there are 7 different genomic targets and multiple drugs that work on those targets, and so it's very rich genomically for being able to follow it and give the patient options when you see those genomic alterations there. There are certain cancer types where, for instance, pancreatic and so on, that there aren't very many drugs out there. It's just difficult, sarcomas, it's just a difficult cancer. Aren't very many genomic targets that are there yet. And so those aren't as well suited. But we still have a fairly high percentage of volume from some of those cancer types. So I'll give you one example. Brain cancer is the one that we have a lower detection rate than almost all the other cancers. That's because there's a blood-brain barrier that prevents DNA from going back through the blood. So in most cancer types, we're around that 85% sensitivity across 40, 50 different cancer types. In brain cancers, we're more 40% sensitivity. But we still have fairly high volume, even though it's less than 1% incidence, just because doing a brain biopsy is so difficult.
What's less than 1% incidence?
The incidence of glioblastoma is very low in terms of cancer types. Brain cancer is an uncommon cancer.
It's only 1%.
Yeah, but it's a much higher representative percentage of our volume, though, because doing a brain biopsy is talk about a difficult procedure. So even if there's a lower chance of seeing something, they'd rather do a blood test first in case you see something, and then then resort to a more invasive procedure.
But that's always been the case, I guess, with brain biopsies.
It's always difficult, but until our test, there hasn't been an option to do anything through blood or an alternative. Basically, it was just do blind chemotherapy or let's do a brain biopsy.
So right now you have about 1,200 oncologists?
Actually approaching 2,000 now.
And how many is it, 10,000 in the country?
About 10,000, yeah.
What's been the biggest challenge, if there's been a challenge, getting this out and more ubiquitous?
It's really awareness. We're a small company, young company. There's a lot of noise. A lot of people talk about liquid biopsies. But as far as we
around for a while, right?
But as far as we know, we're still the only commercial comprehensive liquid biopsy in the market today. Okay. So it's really just getting physicians, getting in front of them, letting them see the data, know that this test exists and you can order it today. We find that when we get in front of a physician, we have much greater than 95% conversion rate. They'll order the test. There's always patients they have that they have no options for, that Guardant 360 can be a good solution for them. But it's really just, you know, getting to their busy schedules and getting in front of them. And so, you know, we've had a very small sales force, but despite that, we've got the 20% of oncologists, which, you know, is pretty fast progress. Yeah.
And it's just in the U.S., or are you also overseas?
No, we're in now over a dozen countries. We announced fairly large partnership with Labco, which is part of SynLab. They're probably one of the largest international distributors outside of the U.S. We'll be covering most of Europe and a large portion of South America with them. We have relationships with multiple distributors in Asia and in the Middle East as well. So we're seeing some good volume from there as well.
When do you see these tests going to your your physician, your primary physician, where you can take an annual and also do a blood draw and know whether you even detect the cancer?
Yeah. I mean, I think, you know, that's something we're definitely committed to in terms of proving up the science and the technology. I think that's the biggest barrier.
When would that be? When's your
You know, I think technologically you could do it in the near term, in the next, you know, year or two, but it's really the clinical studies that are the gating, you know, item, and doing them large enough that, you know, prove out, you know, the technology works. Because I'll give you an example, right? I mean, let's say you have a really sensitive version of this technology that finds these mutations in the blood in a patient. You think you're healthy, and you take this test and you find, you know, these 4 or 5 mutations. So what do you do next? You know, you don't know where the cancer is. You just know that there may be something lurking there. And so we have to think about, you know, just what are the treatment paradigms that we create around that? Do we have an active surveillance kind of mode where we're doing CT scans for that patient, you know, every 3 months until we find something? Or do we just monitor them every 3 months with the blood test and see if those, you know, mutations go up in concentration? So a lot of that has to be formulated before we just create anxiety in patients and so on. So there's certain things you can technologically do today, but, you know, it's kind of also, it has to be, you know, gated and put in check by how we actually use them in practice. And that's some of the things we're working towards.
And do you see this liquid biopsy for anything else beyond cancer?
Yeah, you know, I think you think about it, every organ system in the body is shedding DNA or shedding different, you know, biomarkers that are there. So I do think when we think of sequencing the human genome, we think of that as a static event that happens once in our lifetime. Like, I had my, you know, genome sequenced. I think, you know, over the next, you know, couple of decades, that will change where we're going to have our, you know, genomes sequenced, you know, potentially every year, because we're a multicellular organism. Every cell in our body has a genome that's changing slightly. And not only that, the human genome is only a small fraction of the genetic material in our body. The gut, you know, microbiome, all of that, you know, apparently only, you know, if you only took the human component out, it's less than 1% of the genomic diversity that's in our bodies. Most of it is bacterial and all these, you know, other organisms that are living within us. And so our definition of sequencing the genome will change over time and become much, much more complex. I mean, it's really going to be the difference between, a car phone back in the late 70s or early 80s versus, you know, an iPhone 6 today, just, you know, in terms of what you can do with it and how pervasive it is and how useful it will be.
And do you have an idea of what would be next beyond cancer then?
Yeah, so I mean, I think there are many companies working on infectious diseases.
Would you be working on that though?
We're pretty focused on oncology.
Okay.
You know, it's a $30 billion market by itself, so we have a lot of work to do.
You brought up market size there, and just to clarify, you make Guardant Health is not doing this as a philanthropic venture, so you make money for every test.
So every test we run, we have 4 sources of revenue for most of our volume. You know, most of our volume, we work with private payers, so like your large national payers, to essentially work with them to get paid for the tests that we run. So that's a large portion of our revenue. We also have direct contracts with many hospitals where they pay us, you know, for every test, and they take the risk. We also work, internationally. All of that is directly contracted. And then finally, the largest growing part of our business has been the work we do with pharmaceutical companies, where they're just looking for these patients for these trials and they can't find them. Some of these genomic alterations only occur at 1% frequency in lung cancer patients. So right now they're doing 100 biopsies to find one patient, when we could be doing 100 blood tests in the comfort of the patient's home and finding that patient and putting them into these trials. And so we're doing active screening for many pharmaceutical companies, among other things. And so that's also a large portion of our revenue.
Okay, well, Helmy, we're at 25 minutes now, but I don't want to end it there. The last question is oncology.
Sure.
And why? Why personally for you? Why did you want to solve this problem?
That's a good question. So back in 2011, I went through kind of a little bit of a personal healthcare scare where, I just started feeling really bad, like 50%, had brain fog and, arthritis symptoms and stomach pains and, just a whole bunch of symptoms. And I went to maybe 12 different specialists. I had a CT scan, colonoscopy, endoscopy. There were certain things that were off, but no smoking gun. And one weekend we had, this make your own pizza night, and then, bagels, and we had all these guests. And so I had this basic gluten overload. And even though I had been tested for celiacs and I was negative, my physician, so I broke out into the worst symptoms I had, over this 6-month period. And, I basically found out that for whatever reason I was gluten intolerant all of a sudden, even though I love bread and, it's painful for me now. But I cut gluten out, and within 3 to 4 weeks I started feeling, 90%, approaching 100%. And so, what I realized through that process was, you have physicians with the best of intentions, but they're lacking so many tools. Like I went through this diagnostic odyssey basically where, test after, I probably had 40 different blood tests, and none of them were really that informative. And, I could be, staying, and working on sequencing technologies and trying to make the $400 genome and $200 genome. But what I realized was there's so many basic healthcare questions we can answer today with technology that's sitting on the sidelines that we're not doing because there aren't enough people who have the courage to try to push these technologies into the clinic. And so me and my other co-founder felt that if we weren't going to do it, then who would? And it's definitely something we would have regretted for the rest of our lives if we weren't part of that push to really give physicians the tools they need to, help patients. And what area besides cancer, we think that's the one area when you look at every area in healthcare where we're spending the most amount of money in terms of increases and seeing the least outcomes in terms of improvements there. And a part of it also is I lost my grandmother to colon cancer. She was young. She died at age 40, and so so it runs in my family. So it's definitely something that both me and other co-founders think about and have had personal kind of relationship with. So there are multiple reasons of why we're so committed to this cause.
Well, that's a great reason, a great story. Helmy, thank you so much, and we will see you on February 23rd at Vator Splash Health.
Yeah, really looking forward to it. Thanks, Mamie.
Thank you.